In this cell biology and biochemistry video, we're going to talk about the relationship between mutations and cancer. In the previous video, we focused primarily on the effects of mutations in the coding sequences and how they impacted or potentially impacted amino acids in the final protein. We're going to consider that along with mutations outside of the coding regions in the regulatory regions in this video. We'll also talk about a phenomenon called pleiotropic effects, where mutations in one gene can impact the expression of multiple other genes. We'll consider the impact of mutations in germline cells, those that produce sperm and eggs, versus mutations that take place in somatic cells, those that are just part of the rest of the body outside of the germline cells. And then we'll finally wrap it up by tying in the concept of these various mutations with cancer in general, understanding that cancer is a very broad topic, a very broad set of pathologies. So some mutations take place outside of the coding sequence of a gene. So for example, mutations might happen in the promoter of a gene. And by mutating the promoter, we could either increase or decrease the rate of gene expression, and that will impact the overall phenotype of that cell. We could have mutations taking place in regulatory elements where transcription factors bind, such as operators. We haven't talked about operators yet in this class. You'll get into those in genetics. But an operator is another regulatory binding site close to the promoter that impacts overall transcription rates again. Sometimes those rates go all the way down to zero, and other times they go to full speed or someplace in between. We can get a mutation at splice sites. If you remember when we talked about one of the differences between prokaryotic and eukaryotic transcription is that eukaryotic transcripts need to be spliced generally. There are introns, intervening sequences, that must be removed and exons spliced back together. And if we mutate those splice sites, we may mess up the ability of that pre-mRNA to be spliced into a final functional mature messenger RNA. Translational regulatory elements, so the transcript ultimately has to then guide the process of translation, and we may find a mutation taking place in these translational regulatory elements. So for example, in prokaryotes, maybe the ribosome binding site gets mutated, and that can alter the ability of the ribosome to bind, or translation to happen at a particular rate, or to happen at all. Potentially, you can get mutations in intergenic regions happening between genes, and in many cases, those regions are acting as essentially spacers between genes, and they don't appear to have dramatic impact on the gene expression at all. We could think of those almost as neutral mutations. So mutations can happen outside of the coding sequence. So mutations can happen virtually anywhere within the DNA. Now, I also want to introduce the idea of a pleiotropic effect. Hopefully we understand now that some gene products are essential for expressing other genes. So for example, in this image, if gene X codes for a transcription factor, if gene X gets mutated, either to impact increase expression of the transcription factor, decrease expression of the transcription factor, or even alter the sequence of the transcription factor and therefore the way that it folds and behaves, then all the genes that transcription factor is responsible for helping to regulate the expression of, in this case, genes A, B, C, D, and E, their expression is going to be impacted because now this transcription factor doesn't behave the way it was supposed to behave. So a single mutation in one gene can have multiple downstream effects on the expression of multiple other genes, either increasing their expression, decreasing their expression, shutting it down entirely, a variety of things that we can think of. We call these pleiotropic effects. And so I want you to have that in mind as we move forward thinking about genetics, because it's important to understand how potentially broad of an impact a single mutation can have. All right, another thing we want to consider is where the mutation takes place. Does the mutation take place in a somatic cell? Okay, on the right-hand side, we've got somatic mutations, right? These are the body cells, the ones that have nothing to do with sperm and eggs. If a somatic mutation happens early on in development, like we see in this embryo here, then all the cells that are derived from that mutated cell are going to carry that mutation, but then none of the gametes will carry that mutation because, for example, in this case, they're showing mutation taking place in this region of the embryo, claiming that it impacts this region of the body, and the sperm and eggs don't originate from this region of the body, and so it's not going to be inherited by the next generation. However, in the germline refers to the cells that are producing sperm and eggs. In this case, they're showing a male, and so they're showing that it's happening in the sperm cell. And so this individual, his sperm cells are going to carry the mutation. The mutation then will be in half of his sperm cells, and therefore half of the embryos that he's capable of forming with his future mate are going to carry that mutation as well. So you can see the impact of a mutation in the germline cells producing the sperm and eggs can potentially be a much greater impact. It could even be a devastating impact, but it's going to have a much bigger impact as opposed to a somatic mutation, which is going to be localized physically as well as to a single individual. So what does all this have to do with cancer? Cancer refers to mutations that it refers to a cell cycle that's out of control. We've talked about the cell cycle. Cells don't always need to divide, and when a cell begins dividing out of control, not holding back division, we end up with cancerous growth. That's what cancer refers to. Lots of different types of cancer. We're going to look here at a form of lung cancer and think just a little bit about what goes into it. So here's an individual, and in her the epithelial tissues lining her bronchial tubes, we can see a single cell that has become cancerous. We'll talk on the next slide about how that happens, how one cell picks up the appropriate mutations to become cancerous. But from that one cell then, it's going to divide out of control and form what's called a benign tumor. But if more mutations are picked up, that benign tumor can become aggressive, and then we call it malignant. And it begins actually invading further tissue, potentially metastasizing, which means some of these cells breaking off into the bloodstream, lodging elsewhere, and forming new tumors elsewhere throughout the body. This is potentially a path that a single cancerous cell can cause and can take. If we can remove the tumor while it's still benign, then we're able to block it from becoming malignant, from invading tissue and causing damage, and ultimately from metastasizing. A lot of our cancer therapy is geared towards attempting to stop the tumor from getting anywhere beyond this early stage. Now, how did this initial cell become cancerous? Well, cancer, like I said, is loss of control of the cell cycle. And there are a set of genes that we've talked about before in less specific terms that control the cell cycle. And if any of them get mutated, then potentially we can have cancerous growth on our hands. The first category are what we call proto-oncogenes. An oncogene is a gene that leads to cancer. Think oncology, right? Onco refers to cancerous. Proto just means pre or early. So these are genes that are normal, non-mutated. They're functioning in cell division in one form or another. So for example, a growth hormone, gene encoding for maybe epidermal growth factor, or potentially signal transduction proteins that are involved in helping to tell a cell when it's time to divide. These would be proto-oncogenes when they're healthy. If they pick up mutations in just the right places, they get converted to what we call oncogenes, and now all of a sudden they're telling the cell to grow and divide all the time. Another example, and these are really the two most important categories, the other really important category of potential cancerous genes are what we call tumor suppressor genes. Tumor suppressor genes, under normal, non-mutated conditions inhibit cancerous growth. In other words, they're checkpoints. You remember we talked about checkpoints in the cell cycle. For example, the G1 checkpoint has a set of proteins that hold back the cell from moving into S phase, where DNA replication takes place, until everything looks right. A real common example of a G1 checkpoint protein that is a tumor suppressor is called P53. Something like half of all human cancers include a mutated P53 gene. So we've got these cell cycle checkpoint proteins that if they get mutated, they're tumor suppressors, but if they get mutated, they no longer suppress the growth like they're supposed to. Typically, cancer is going to involve 6-7 mutations easily in a single cell in both proto-oncogenes and tumor suppressor genes. It takes a whole set of mutations ultimately for a cell to begin growing out of control. And really, this is the primary link between these DNA mutations and cancer. So let's try to hit some of the highlights from this particular lesson. We talked earlier about how mutations can occur within the coding regions and how that impacts the final proteins. We need to understand that mutations can happen outside of these coding regions and have dramatic impacts as well. Some mutations can have pleiotropic effects, so a single mutation in one gene can impact dozens of other genes. Mutations in the germline cells, those that produce sperm and eggs, are heritable and therefore can potentially have major impacts compared to those in the somatic cells. But in the somatic cells, multiple mutations in key genes, in proto-oncogenes and tumor suppressor genes, can accumulate and result in cancerous growth in that material. And that cancerous growth can begin benign, but it can become, through multiple mutations, malignant, where it begins to invade nearby tissues, potentially metastatic, where it then spreads throughout the body and results in tumors throughout the entire body. So spend some time on this video, learn the terminology, make sure the concepts make sense. I'd recommend you have your textbook open as you're going through all of this to make sure you can fill in all the blanks. Thanks for joining me.